Good morning, everybody. I had a boss once who said, if you're going to hoot with the owls, you have to sing with the larks. So I hope everybody's feeling refreshed after the excellent hospitality last night. Because you'll need to sit up and pay attention for this next session, because we're going to hear 7 stories from 7 groups of colleagues and partners, very special to MMV, as we look back in the past 20 years and celebrate some of our key achievements. With that, I'd like to invite my colleagues to the stage already to take up the seats. Groups 1 and 3, please, if you come from your left, and the rest to come from the other side. So over the last 20 years, MMV has had the pleasure of working with over 400 partner organisations in 55 different countries. So that, in terms of individuals, must be thousands of people that have worked with MMV. And today we'll see a little bit more of the tip of the iceberg that represents the achievements of MMV to date. But as you can imagine, it's still a lot to cover. Our first story begins back in 2003. 4 years after the formation of MMV. At that time, a medicine called Coartem, developed by the Swiss pharmaceutical company Novartis, had become the artemisinin-based combination therapy, or ACT, of choice for treating uncomplicated malaria in adults. However, there were no high-quality ACTs formulated specifically for use in children, not a single one. And we are talking about a disease that predominantly affects children under the age of 5. Reaching those at-risk and underserved patient populations is at the heart of MMV's mission. And this was the driving force behind what today is one of MMV's longest-standing collaborations. Using its understanding of the reality of treating malaria in the field, MMV worked with Novartis to develop a new dispersible formulation of Coartem that would be safe for use in those most at risk, those very small children. And today, MMV is delighted to announce that since the launch of Coartem dispersible 10 years ago, 385 million treatments have been distributed to malaria endemic countries, an incredible achievement. And the impact of this has been enormous. It's estimated that Coartem dispersible alone has treated over 800,000 children to date, and importantly has paved the way for the development of other child-friendly formulations. I'm now pleased to introduce some of the team behind the Coartem dispersible development, who will now tell us more about this remarkable story. Please will you come up to the microphone.
Good morning. My name is Hans Rietveld, and I'm very privileged to have joined MMV here in April this year from Novartis, where I was part of the core team that launched Coartem. And as we just saw from the animation, back in 2003 when Coartem came into the market, it was not available really in a formulation adapted to children. Mothers were being creative in making their children swallow the adult tablets that were crushed and perhaps sweetened with a little bit of honey or fruit. But it wasn't optimal. And so MMV recognized that gap and approached Novartis and said, we are here to help. Let's work together. And so soon after, indeed, the first collaboration agreement between MMV and Novartis was drawn up, and work started. And I remember it wasn't quite straightforward to get the right formulation in place. But when that was the case, the next challenge arose, because we weren't able to demonstrate bioequivalence between the new dispersible tablets and the crushed tablets. So that called for a major pivotal trial to compare the two formulations. And so as we worked together, the collaboration between the two organizations naturally extended. I remember working with colleagues to develop patient-friendly packaging that was award-winning. We also set out to test community health worker training materials that then supported, indeed, the launch of Quartum Dispersible in 2009. And then, indeed, we spent many days together, including with Jaya and others in the team, to prepare for the launch of Quartum Dispersible. And that was a wonderful collaboration, and I would say that was and is a great example of the power of partnerships.
Hello, my name is Rebecca Stevens, and I head Global Health Partnerships at Novartis. I worked with Hans and with Jaya at the time when we launched Quartum Dispersible. The clinical development of Quartum Dispersible at Novartis was a huge team effort. In addition to Hans, the team also included Anne-Claire Marast, who is also now the Senior Medical Director at MMV, and many, many more colleagues. For me, the story of Quartum is a very personal one. As a young child growing up in Sierra Leone, Freetown, Sierra Leone, I often had to take very bitter malaria tablets whenever I had malaria. More than the illness itself, taking the bitter tablets was very much of a horrible, horrible experience. And just for the anecdote, we were 5 children. So whenever 3 or more of us had malaria, just imagine my mom having to get us to take bitter tablets. I think for her, it was also a horrible experience. The Novartis Institute for Tropical Diseases arrived at the product we launched in 2009. By the following year, 52 million treatment courses had been shipped to 33 countries. Really a landmark moment for antimalarial drug development. For the past decade, Quartum Dispersible has been treating vulnerable children in malaria-endemic countries every single day. For me, I feel a double amount of pride when I hear these numbers. First, as a Sierra Leonean and as an African, I am proud to have played a role in helping to develop a medicine that has a positive impact on the lives of many African children. And secondly, I feel an immense amount of pride working for a company that sees the development of malaria medicines suitable for children as a priority. Thank you.
I'm slightly shorter. Okay, so hi, I'm Joya Banerjee. I've been with MMV since 2006. That's a very long time, and time enough to actually see this wonderful project develop. As Rebecca said, Quartum Dispersible was launched in, 2009, 10 years ago, after regulatory approval from Swissmedic. But that was the first step, because as Hans mentioned, a lot needed to be done afterwards, actually. Novartis and MMV had to ensure that Quartum Dispersible was used rationally. So that meant administering the drug, which was pretty complex. It's a complex process. And we had to commit to supporting local healthcare workers and communities to ensure, proper use of the medicine, and thus maximize the benefit to young patients. So we worked closely with Novartis to ensure the instructions for use were self-explanatory and easy to follow. It made it easier for caregivers to with lower literacy and transcend language barriers. So we used iconography and infographics in our communications, as Hans mentioned. It was a wonderful experience, actually, to work with the Novartis team on that. And good to say that the drug was then well received, and the approach of infographics and easy to understand pictorials was also well received. What's exciting to know is that our work so many years ago has helped over 825,000 children receive the right dose of this antimalarial.
to the Coatem team. So around the time that MMV and Novartis began to work on Coatem dispersible, another major problem needed to be addressed, and that was the issue of an empty anti-malarial drug pipeline. No new compounds coming through. So to reinvigorate anti-malarial drug discovery, MMV partnered with global pharmaceutical companies to explore their chemistry libraries to identify new compounds to replenish that pipeline. But screening compounds in the early 2000s was relatively slow, and so MMV supported the establishment of validated high-throughput technology to enable a better and faster screening process. Since then, MMV's translational science platforms have catalyzed the discovery of 30 novel drug candidates and continue to evolve in response to the market need by driving and adopting technological innovation. Some of the science involved in those early, exciting years will now tell us more about MMV's first foray into the discovery of new chemical entities. Didier, Javier, Elizabeth, please will you join us at the microphone?
Hello, my name is Elizabeth Winsler, and I started working on malaria and drug discovery when I was at the Genomics Institute of the Novartis Research Foundation in about 2004. Simultaneously, I ran a lab an academic lab at the Scripps Research Institute, and I'd been developing high-throughput ways of studying malaria parasites. At the time, I was mostly interested in using functional genomics to find new targets. And given my lab's expertise in parasite biology and the Institute's high-throughput phenotypic screening capabilities, which in the early 2000s were unparalleled, we wanted to see if we could bring these two capabilities together to try to replenish the anti-malarial pipeline. We set about developing inexpensive, simple, and precise assays that might predict if a compound could be developed into an effective anti-malarial medicine. At the time, the late Ian Bathurst was leading the project from MMV, and working together, we were able to show that this approach would likely yield molecules that could be rapidly developed into new drugs. I was very impressed by the passion and expertise Ian Bathurst brought to the project. He became a great advocate for phenotypic screening and encouraged others, such as Vicky Avery, to get involved.
Hello. My name is Javier Gamo, and I have been working at GSK in discovery since 2000. And just like Elizabeth, we were looking at how we could quantify the effect of a drug on parasites. In Tres Cantos, it's near Madrid in Spain, we were developing a methodology to allow us to accurately measure the killing rate of a given drug and shed light on its possible mechanisms of action. At that time, we were investigating pyridons as electron transport inhibitors, and we realized that despite all the published data, these inhibitors were not actually effective at killing the malaria parasite. In addition, we found notable differences between widely used standard antimalarials in terms of killing rate. Together with MMV, we realized that this could have translational implications and could help with the prioritization and clinical development of future antimalarials. Most recent antimalarial candidates have been analyzed through our assay in collaboration with MMV and GSK, which give us great satisfaction. In fact, the parasite reduction ratio has become an important part of the data package of new compounds and plays an important role in portfolio prioritization decisions.
Good morning, everyone. I'm Didier Leroy, Senior Director in Drug Discovery at MMV. Before we miniaturize the red blood cell assay and standardize the way we describe the effect of a drug or compound on parasites, MMV had relied on project applications to identify new projects and novel compounds. High-throughput screening technology made the cost of screening projects competitive. It made collaboration with pharmaceutical companies viable, and we have had the privilege of working with an impressive range of partners over the last 2 decades. Working with standardized assays that generate comparable data sets has really allowed us to operate in the way we do today, to make transparent decisions about which compound or chemical series to prioritize and to push forward the most promising drug candidates. To date, MMV and partners have developed and validated more than 15 different assays throughout the entire life cycle of the parasite. 7 years ago, we published a fingerprinting exercise whereby around 50 molecules representing the chemical diversity of the various classes of antimalarials were used to validate these assays. Being able to reconstitute in vitro the complexity of the life cycle of the parasite was the basis of our current process for profiling our portfolio compounds. A key complementary component of discovery was to couple in vitro assays with tailor-made in vivo pharmacology models as an essential path towards translational activities prior to first-in-human studies. As we speak, with the help of TAD, a modern drug discovery company in Bilbao, we are about to reconstitute the entire life cycle of Plasmodium falciparum in humanized in vivo models at an industrial level, and this is something that represents a real breakthrough. Today, we continue to explore ways to harness new findings and new technologies to improve our discovery and translational platforms whose fundamental bases were established 10 years ago by our deeply missed visionary colleague, Jan Bathurst. Thank you.
Thank you. So we've heard about how MMV has helped to drive the development of new child-friendly medicines and to replenish the pipeline. But as we all know, it can take up to 10 to 15 years to bring a new drug to market. In 2001, MMV and Shinpoon, a Korean pharmaceutical company, joined forces to develop and deliver Pyramax, a powerful and convenient alternative ACT. This project is a fabulous example of how MMV has helped bring about a valuable therapy that may never have come to market without a partnership approach. Members of the Pyramax project team will now tell us more about the history of the drug's development.
Good morning. Hello, my name is Isabelle Borghini, and I have been working with MMV since 2006. 2006, it was the time when we were designing and planning for the phase 3 clinical trials of Pyramax. It was a large program of 5 phase 3 trials. 5 phase 3 trials. So in Asia, in Africa, in adults, in children, in Plasmodium falciparum, and in Plasmodium vivax malaria. So quite a challenging and ambitious program, and it came with challenges. Remember, in 2006, we were 22 people at MMV. And one of the challenges that I would like to tell you about was actually a very nice one. Our protocols ask for the patients to be hospitalized for the first 3 days. And I'm sure that Michael Ramarter or Professor Guy, you will remember this. We sometimes had difficulties finding our very young patients for the 3rd day because they had run out of the hospital to play football, because they felt so much better so quickly. So the symptoms really were annihilated very fast with this new drug. So that was a nice challenge to have. Anyway, so why Pyramax? Well, in fact, in 1999, it was the WHO who approached Shinpoon Pharmaceutical, together with Professor Larry Fleckenstein of University of Iowa. Larry is here with us today, to explore whether it would be possible to develop a new ACT combining pyronaridine with artesunate. This was to offer options for the frontline treatment of malaria, and also in anticipation of the development of possible resistances. And Shinpoon, despite being mainly focused on manufacturing and distributing generic drugs, their late Mr. Chairman, Mr. Yong-Tek Chang, accepted the project wholeheartedly. And in 2001, MMV joined the project, together with Shinpoon and Larry Fleckenstein from University of Iowa, to contribute its anti-malarial drug development expertise. So I have personally had the privilege of meeting Mr. Chang on a number of occasions in Seoul, and he was really an inspirational man whose vision, we can say, has directly improved the lives of millions of people. And I know from talking to him that Piramax was one of the products of which he was most proud. And we are really delighted that his son, Wonjun Chang, who was with us last night, is also passionate about Piramax and will continue our partnership, developing and even facilitating access to the product in malaria endemic regions like Africa.
Thank you, Isabel. Good morning, everybody. My name is Adam Aspinall. I joined the Access and Product Management team at MMV back in 2013. In fact, not long after the first European Medicines Agency positive opinion for Piramax. And at the time I joined the team, it seemed to me that most of the hard work had actually been done. However, as we know, life's not always quite that simple. And in fact, the regulators did still have a few questions, specifically around hepatic safety and repeat dosing. So, in partnership with Shimpu and other partners, we embarked on a very ambitious program, huge-scale clinical studies, first with the Wanakam network in West Africa, and then with the Cantam network in Central Africa, and also a study site in Côte d'Ivoire, to generate data to address these concerns. And in doing so, to bring Piramax to more patients who need it. And as you all know, evidence-based medicine is just that, is evidence-based medicine. And without evidence, we can't move to policy. And without policy, we can't move to access and getting the drug to the patients that need it. And in that case, nobody benefits. And gathering this evidence involves not only investing heavily in capacity building, but also a multi-partner, multi-country approach. And as we move towards launching new combinations in the future, the experience we've gained with Piramax is going to put us in a great place.
Right, good morning. I'm Bernard Zogudu from Kenya, and I've been hanging around malaria since 1991, when I became a doctor. And currently working with the Kenya Medical Research Institute and also Strathmore University with the main thing to optimize treatments for malaria and seeing it to the end. The data generated by MMV and partners on Piramax is important for us at the front line to help policymakers make the right decisions, to inform policy change, letting us know whether the new treatments or ways of deploying them will be effective as we move towards multiple first line, and also allowing us to achieve the evidence-based medicine to the health workers and make them make effective treatment decisions, because I think that is our biggest problem currently in most of the endemic countries. And last, it is needed for consensus building among stakeholders so that we can all understand what options, both medicines and deployment strategies that we need, because I think this time we are lucky. It is not the drugs, but the strategies that are going to matter so that we make them available and decide on which one works best for our populations. And I think that's what we are really going to work on greatly. Adding Piramax with its huge data package to the antimalarial treatment options we have in our country gives the healthcare professionals more choice, but also more headache, because we have to tell them the right thing to do. And this is important when treating patients of all ages at a community level. And I think this has been our biggest nightmare, that elimination of malaria will require community participation, and we need to make sure products are there. Indeed, having multiple first-line options will be the future of malaria treatment. And whether we like it, that is where the future is. It will allow us to treat patients who can't tolerate some therapies by choosing the therapy most likely to be well tolerated by the type of that patient, as well as rotate the use of first-line therapies such as Coartem, DHAPQ, and Piramax to limit the risk of drug resistance developing pretty fast. And I think that's my message to you.
And just finally, to finish off this session, with perfect timing, in fact, for this 20th anniversary of MMV, Just last month, we had some amazing news from the WHO. As you know, at a country level, guidance from the World Health Organization and the Global Malaria Program is incredibly important in helping inform national treatment guidelines and what our colleagues do at a country level. And last month, the WHO issued an information note on the use of Piramax. And what it said, and I can read directly, is that Piramax can be considered a safe and efficacious treatment, ACT, for the treatment of uncomplicated malaria in adults and children weighing 5 kilos and over in all malaria endemic countries. It can be considered for procurement and for also including in national treatment guidelines at a country level. This has been a long time coming, and if that's not worth a round of applause, I don't know what is. Thank you.
Thank you, Adam. That's excellent news indeed. And actually, when I first joined MMV, it was in 2009, and MMV, I can't take any credit at that point, of course, had just successfully submitted the dossier, and I thought, wow, this organisation really means business. But I think, as Adam said, the work's not over just because you have an approval or you've done the submission. And I think it's one of the things for me that stands out about MMV is that we never give up, and we keep going even when the road ahead is tough. So that's excellent news for Piramax, as I said, and for the treatment of uncomplicated malaria. But what about severe malaria, the form of malaria that's so bad that the patients can no longer take medicines and their lives may be on the brink of extinction? Injectable artesunate remains the recommended frontline therapy for severe malaria and continues to save lives around the world. Since 2011, 144 million vials of Artesun, the injectable artesunate product manufactured by Fosun Pharma, have been delivered to patients, treating an estimated 840,000 additional lives versus quinine administered in the same period. As the first WHO pre-qualified injectable artesunate treatment on the market, Artesun is now approved in 33 countries worldwide, and demand continues to grow and additional supply is needed. In 2018, MMV is proud to support a second manufacturer, the Indian company Ipca, in achieving WHO pre-qualification of their product, ensuring supply security for the future. The issue was, though, how was this life-saving medicine going to get to patients who most urgently needed it? We're now going to move to a story that highlights that when the going gets tough, as I already mentioned, MMV and partners pull out all the stops to ensure that the hardest to reach and most affected people get the healthcare they truly need. I'd like to invite Pierre and Elizabeth and Christian to the microphone, please, to tell us the story of the injectable artesunate. Thank you.
So good morning, everyone. My name's Pierre Hugo, and I've been with MMV for 9 years now. And although my current focus is on market shaping activities to support the access and product management portfolio through developing supply and manufacturing solutions, I'll never forget the time I spent working with partners to support the introduction of injectable artesunate in DRC, a country with the highest burden of severe malaria and the highest death rate of malaria in the world. It was in 2011 that MSF, Médecins Sans Frontières, released a policy paper advocating for countries to switch from quinine to injectable artesunate in order to save more lives from severe malaria. However, we already had injectable artesunate, product Artesun, which we'd worked with Fosun Pharma, and I think we've got our Fosun representatives in the room all the way from Shanghai, so very privileged to have them join us and lovely to have you here, to achieve WHO pre-qualification so that it could be procured with donor funds. So that was definitely groundbreaking to be able to have a life-saving drug available for treatment that was now pre-qualified. So in 2012, we set out to successfully bring injectable artesunate in one of the hardest hit countries in the world. So no mean task. It was a big challenge. We knew that it would be an uphill struggle to get severe malaria treatments to patients, but with the help of our partners, Cis Tropical Public Health, President's Malaria Initiative, the National Malaria Control Program in DRC, we succeeded, and you'll now hear the story of what that partnership involved.
Thanks very much, Pierre. Good morning, Geneva. My name is Christian Lengeler. I'm an epidemiologist with the Swiss Tropical and Public Health Institute in Basel, in Switzerland. I've been involved and participated in the work of MMV for over 10 years now. One of the great privileges and joy of my career was to be the chairman of the Access and Product Development Partnership for 6 years. And then in 2012, as Pierre said, we embarked on this study called Matthias, trying to compare injectable artesunate with injectable quinine in Congo. It was a really very interesting and very challenging study. But basically, Matthias confirmed what we knew from clinical trials, that injectable artesunate was really much more, was very safe, much more practical, and really led to better clinical outcomes. Really, the work in Congo was also opening, I think, the eyes to all of us that it's actually incredibly complex undertaking, both treating malaria, preventing malaria, and then also organizing that very complex treatment pathway for severe malaria. It's going to be a very, very serious challenge to deliver on a promise to eliminate malaria by whatever date it is. I think in comparison, putting a man on the moon is a pretty straightforward operation. So also in the time at APMAC, we had many discussions as whether MMV embarking on the issue of access and product stewardship down the line was not mission creep. I think a few years later, and as we celebrate the 20th anniversary of MMV, I think there is no doubt that it's part of the mission. It makes MMV a better operation, a better institution, and leads to really amazing results. Thank you.
Hello, my name is Elizabeth Chizema, and when I joined the MMV Partnership Network, I was head of the National Malaria Elimination Center at the Ministry of Health in Zambia. And I'm going to tell you about how rectoartesunate is changing the way severe malaria is managed in our country. You may be aware that Zambia recently revised its national policy to align with WHO recommendations, replacing quinine with treatments such as injectable artesunate and with rectoartesunate for the pre-referral management of severe malaria. To make this successful, we needed to train a lot of people on how to use the new therapies. So the National Malaria Elimination Center in Zambia, with MMV and TransAid, which is a UK non-profit organization, set out to do just that. By working with multiple partners, we've been able to introduce and increase access to quality-assured rectoartesunate, as well as scale up and strengthen existing services and reduce the disease burden. By the end of 2019, with the help of the various organizations such as PATH, the Global Fund, the Rotarian Malaria Partners, as well as US President's Malaria Initiative, we'll have trained over 14,000 community health volunteers, so that now we can proactively diagnose and manage cases of severe malaria at the community level, rather than waiting for cases to present at the district health facility. This, in turn, reduces the pool of individuals who can contribute to malaria transmission. Giving rectoartesunate to children with suspected severe malaria allows us to buy time while the child is being transported to the district health facility. This extra time can mean the difference between life and death. Almost 100% of the children treated with rectoartesunate for suspected severe malaria in our pilot study in 2 districts reached the health facility in good time. And all thanks to innovative implementation of bicycle ambulance and the team approach. Ultimately, for maximum coverage, we hope that initiatives like this one will one day be present in all the 114 districts of Zambia. And I also hope that our experience will inspire the other African countries to consider similar projects and forge new collaborations.
Thank you. So, as we've heard, it can take a long time and a lot of data and plenty of money to bring a medicine from the lab to the patient. To expedite drug development process, MMV and partners have adopted innovative ways to answer important clinical questions and de-risk the development of new combinations for malaria treatment. Since 2012, human volunteer infection studies have transformed the anti-malarial drug development pathway, accelerating the development and the prioritization of new medicines. Let's learn more about this groundbreaking new platform. Jorg, James, please will you take the microphone?
Good morning, I'm Jorg Mörle. I joined MMV in 2005, so it's really great to see so many colleagues and moreover friends who have come today to celebrate the 20 years anniversary. Have you seen from the video, but also from the presentation by Elisabeth, Javier and Didier, that the pipeline started to fill up in 2010, and we needed a way to really study and characterize these new drug candidates, whether they actually can treat people who have malaria, alone and in combination, without placing a huge burden on the patients and without risking the life and the health of these patients. Also helping to process faster, to do these studies faster, just to work with that pipeline to weed out the compounds who are not good enough and prioritize the compounds who are promising to go into late-stage development. We used to conduct studies, the first studies in patients, in adult patients, in Southeast Asia. However, since it was the first activities where we could go into patients, we only had limited information, and the study design did not allow us to observe these patients for a long time. So after giving these patients who came in ill the investigation, the new drug, after a very short period, we were obliged by the protocol and by the ethical committee to give a standard of care, COARTEM. So that also allowed us only to observe for a short period of time, about 24 to 36 hours, how the drug works. So we got information on how fast the drug can kill parasites, but we did not get the information whether the drug can cure the patients, nor what are the critical concentrations that are necessary to kill the parasites in humans. So we had a full set of phase 1 studies, we had good information, but even the first studies in patients did not give us a lot of information. So there was still a gap, and these studies took a long time. We started to look around what are other options, what are better study designs, how can we do that better, and we discovered old news, malaria challenge study or volunteer infection study, so infecting healthy humans with malaria and then study the effect of a drug. Infecting humans with malaria was not a new concept, it was actually developed 100 years ago to treat syphilis, because the fever episodes killed the syphilis-causing agents treponema. So there was a lot of information on infecting humans with malaria, in this case to treat another disease, but later on also these infection studies were done to discover or to characterize new drugs such as primaquine, mefloquine, and malarone, atavacrone, proguanil. So we were looking around how we can adopt these challenge models to study new drugs, and that's how we encountered James McCarthy, and he will tell you about the collaboration.
Thank you very much, Jorg. My name is James McCarthy. I'm from Brisbane, Australia. It was back in 2010 when, on holidays, I received a phone call from Jorg's compatriot asking me whether I was interested in doing volunteer infection studies with MMV. Initially, I was very surprised, but then thinking and talking about it more, it became obvious that this was a fantastic opportunity to get an early read on the effect of novel antimalarials. So with the support of MMV at the Queensland Institute of Medical Research, we developed over a period of 3 years a fully functional clinical trials unit able to collect the appropriate very early safety and efficacy data using a sensitive PCR test to actually measure the parasitemia in healthy volunteers who'd been experimentally infected with malaria. So since that time, we've expanded our capacity to do these very novel research projects, including other parasites such as Plasmodium vivax, and more recently using the drug-resistant K13 parasite that has now become an increasing concern throughout the greater Mekong sub-region. For me, the highlight of my collaboration with MMV has been this link between academia and industry and the PDP. What a wonderful way to do very important translational research, develop drugs, and also really make fundamental understanding about the biology of the parasite and how the human host reacts with that parasite. And I think this is a really great example of how MMV has worked with partners and really been incredibly flexible, enable everybody to complete these innovative studies so that we all reach the objectives of developing new drugs for malaria, but also understanding the biology of this otherwise lethal disease. So right now in 2019, we've undertaken studies with over 16 drugs with more than 350 healthy Australian backpackers, none of whom have come to grief. And we're really excited to be able to continue this work to work towards the drugs that are now emerging from the drug pipeline. And as I said, we have a number of really exciting projects, including working with the K13 mutant parasite. So thank you very much.
Thank you, Jorg. Thank you, James. So our story now moves from the outback in Australia to South Africa, and the moment when MMV embraced an opportunity to connect with innovative research groups, share knowledge and expertise, and expand discovery and research partnerships into the continent of Africa. In collaboration with the University of Cape Town, or UCT, and its partners, MMV has helped to empower African institutions, scientists and industry to join the fight against malaria and be part of the solution to one of the continent's greatest health threats. Members of the MMV 048 team are here to tell us more. Christina, will you bring everyone to the microphone, please?
So I'm going to disrupt technology and try and kiss the microphone a little bit. So I'm Kelly Chibale from the University of Cape Town. I actually first met Tim Wells on the 17th of July, 2008 at 6:24 a.m. at Terminal 1 of Heathrow Airport in London, before the word Brexit was even invented some time ago. So that meeting at the Costa coffee shop on the left-hand side of Terminal 1 was very, very instrumental. It was actually a turning point in my career, if I can put it that way. It really soon became evident that Tim and I had a lot in common. We still have a lot in common. That's why we still get along. And I think the thing that really brought us together was the vision to think about building capacity in Africa, starting with malaria and see how down the line, whatever lessons we learned from malaria would be relevant to other diseases as well. So the collaboration really started based on a shared vision to energize and advance drug discovery in Africa, empowering the next generation of scientists. I think we heard from David Reddy at the beginning when he was talking about how elimination may not be realized in our lifetime. So we've got to prepare the foundation and the groundwork for the next generation. And so our part in this is to train the next generation of African scientists that can really participate in solving the malaria problem. So this actual program started really with a discussion on how we could use the malaria project to build capacity in Africa. The opportunity arose when MMV, working with Vic Avery, who I think I saw at the party yesterday, screened a commercial library from a company called Biofocus, which is now part of Charles River. And this is a library that was screened by Vic Avery to identify starting points that we then put into a formal program that started in April of 2009. Of course, at the time, based on the discussion we had with Tim, of course, the only thing that was available in my lab at the time was chemistry. And the discussion was how we could tap into the MMV network to access the other platforms that we didn't have in South Africa. And of course, that's how then working with Sue Sherman, who again I saw last night at the party, in Monash, providing the drug metabolism aspect of it, and also partnering with Sergio Whitlin as the Swiss Tropical and Public Health Institute, providing the pharmacology and parasitology aspects of the program. Importantly with this program was the appointment of Mike Whitty. Mike is now the chair of ISAC as the mentor, not just to mentor me, but also to mentor the project team. This was really critical in ensuring that we can really bring best practice to the program based on learning from people who have done it. In addition to all the bits from Australia, from Sue Sherman, parasitology from Swiss EPH, and Mike providing the mentorship, was access to additional chemistry resources through Syngene, which is a CRO based in India. So when I look back at the last 10 years of working with MMV together, it's really through this partnership that we've managed to really create the first drug discovery center in Africa with all the platforms put together that we didn't have when we began the collaboration. And as I speak to you now, we have a fully integrated drug discovery center. And it's thanks to MMV and to Tim Wells for that vision of using the project to help us build capacity.
Thanks, Kelly. Hello, everyone. My name is Cristina Donini. I've been at MMV for 7 years now, and I started the first in-human trial of MMV-048 back in 2014 at UCT. First-in-human trials are the most challenging type of clinical trial to run because you must adhere to very stringent regulatory standards to ensure patient safety, and in addition, the quality of data, of any data output. To perform such a trial in Africa was thought to be impossible, and as David mentioned, we think that we could achieve the impossible because you need the infrastructure to handle what is unexpected. There are so many unknowns about the new drug that has to be handled. Thanks to the collaboration between MMV and Kelly's group at UCT, and also our joint desire to continue building drug development capabilities in Africa, we thought that this one was a huge opportunity. So working with our partners and the backing of our donors, we carried out the first-in-human trial with the new anti-malarial in Africa, together with Professor Karen Barnes' group. Karen is not here, I think, today. And on the strength of this positive experience, we decided to continue and repeat the capacity building exercise by creating two new clinical research centers in Ethiopia. And this one was a massive concerted effort, but we succeeded. And Rezika and Daniel will walk us through this. Rezika.
Hello, everyone. My name is Rezika Mohammed. I'm based in University of Gondar, in northwest part of Ethiopia. And in 2017, after following the successful completion of the first in-human trial that Christina mentioned, we started a phase 2A clinical trial of MMV-048 at 2 sites in Ethiopia. So as one of the two principal investigators, I worked closely with my colleague, Dr. Daniel, and other local scientists to implement the study. And it was the first few subjects. It was a very stressful project in the beginning. Then ultimately, it was exciting to see patients successfully treated with the new drug. So in terms of the local capacity building, the trial has brought a huge benefit, and now we have a new infrastructure at the health center and a new equipment which meets the good clinical practice guidelines, recommendation, and we have now a better trained staff than we had previously. Thank you so much.
Hello, my name is Daniel Ilma. I'm from Jimma University. I'm working in the opposite direction with Razika in the southwest of Ethiopia. As we heard from Primax, that high-quality evidence is essential to support the uptake. And always evidence generations requires trained personnel and also infrastructure. So for Jimma sites, we were very excited when we're running a phase 2A study for the first time in Jimma. And we are very grateful and for MMV for supporting us in building new infrastructures and also provision of the training. I'm also more excited that our site staffs and the research staffs in Jimma University are now highly motivated in fighting against malaria. And they are they are using the training not only for the study purpose, but also they are making sure that for the capacity development in the surrounding and improving the health care in the surrounding health facilities. And I think empowering African researchers and also institutions to come up with their own health solutions for the health challenge is very essential to come up or to reach to the target. And I hope our experience will inspire more new collaborations and also more efforts in the capacity building. Thank you.
Thank you. For our last short presentation, it's time to turn our attention to a less well-known parasite, Plasmodium vivax. We all know that P. falciparum is the big killer and is always associated with the headlines, but the burden of relapsing P. vivax malaria is often neglected. The relapsing malaria form affects 7.5 million people and threatens 2.5 billion. That's a third of the world's population every year. And I know because I checked it last night. For more than a decade, MMV and our pharmaceutical partner GSK have worked together to co-develop a new drug for the treatment of relapsing malaria due to P. vivax. And in 2018, they shepherded tafenoquine, the first new therapy for relapsing malaria in more than 60 years, through the tough regulatory process, leading to approvals by 2 stringent regulatory authorities in the US and in Australia. But tafenoquine is no simple therapy. It requires 2 suitable point-of-care diagnostics before it can be launched and administered. One for the confirmation of the P. vivax infection, and another to check for a deficiency in an enzyme, which shall be known here as G6PD. Here are members of the tafenoquine team to tell us about this important and very much needed new treatment. Alison Elodie, please.
Hello. My name is Elodie Jambert, and I've been at MMV for 3 years, joining the tafenoquine project at the end of 2016, right before we received the phase 3 study result on tafenoquine. As John mentioned, for 60 years, the only treatment available for Plasmodium vivax was primaquine, a treatment administered once a day for 14 days. As you can imagine, this long regimen led to issues with patients' compliance. So the global community sought a cure that would require fewer doses, ideally a single-dose cure. In 2008, MMV and GSK embarked on a program to find the right tafenoquine dose and a phase 3 study population to get the drug approved and to market as a radical cure for the prevention of relapse of Plasmodium vivax. After 9 years in clinical development and demonstrating its ability to cure after only 1 single dose, tafenoquine was approved by both the US FDA and the Australian TGA in 2018. And I'm very pleased to announce that the Brazilian Health Regulatory Agency has just granted this month a marketing authorization application for single-dose tafenoquine, truly a landmark moment. And the regulatory submission is ongoing in other malaria endemic countries. Our partner from GSK will now tell you about the diagnostic tools that were needed to accompany tafenoquine. Thank you.
Hello. My name's Alison Webster, and I've been part of the tafenoquine team at GSK for the past 10 years, which seems to have gone in the blinking of an eye. And it's great to be here with friends and colleagues celebrating what we've achieved together. I just first would like to acknowledge my colleague, Jörg Peter, or JP Klein, who can't be here with us today. And he was heavily involved from this project from the very beginning, and we're absolutely indebted to him for his dedication and his committed leadership of the project. As Elodie just mentioned, specific diagnostic tools were needed to accompany the launch of tafenoquine. And the reason for this is because both primaquine and tafenoquine, which belong to the same class of compounds, the atamine quinolines, can cause hemolysis, a type of anemia, in individuals who are deficient in the enzyme G6PD. And we were very clear from the outset that launching a drug for relapsing malaria would need an as yet unavailable, affordable point-of-care diagnostic test for G6PD deficiency. And we would need experts in diagnostic development to take the lead on this. And we spent a long time in the project team trying to work out how to get this initiative moving. So to help identify patients eligible for treatment, MMV's partner, PATH, supported the development of a quantitative point-of-care G6PD diagnostic test that's been approved by the expert review panel for diagnostics and in many P-Vivax endemic countries. So now that tafenoquine and the G6PD diagnostic tool are available, we're busy generating the evidence to support their use both in rural and in urban settings before tafenoquine's made more widely available. And we truly hope that tafenoquine will deliver both individual and public health benefits in terms of both reduced relapses and also reducing onward transmission of P-Vivax. And that will help us take another step forward towards the eventual elimination of malaria in P-Vivax endemic countries. Thanks.
Thank you, Alison. Thank you, Elodie. So that brings a close to this first session, and it's a very inspiring collection of achievements. People sometimes ask me if I miss the science, having left a career in science, I guess, to pursue business development. But after listening to all this, how can I miss it? Because I'm still absolutely immersed in it. And the one great thing about business development is, although I do miss the detail, getting into the detail, but you get the chance to work with every single one of these groups. And I've had the privilege of doing that in the last 10 years at MMV. And I've had the great pleasure of sharing with you some of those stories in this session. I think you'll agree that together they've shown that partnerships really do make the impossible possible. So thank you again to everyone on stage for sharing your stories, and thank you to all of you for listening and for the role that you've played in these achievements. I mean, there's many, many more people who've been involved in every single one of these stories that we couldn't mention in person today, but you'll have the chance to do that throughout the day. I would like to invite to the stage now MMV's Chief Scientific Officer, Tim Wells, and the Executive Vice President of Corporate Affairs, Andrew Lucar, to give some closing thoughts and remarks on this session. After we come back from coffee, the real Andrew Jack from the UK's Financial Times newspaper will chair the next section, working towards a common goal, malaria and the 2030 Agenda for Sustainable Development. But first, Tim, Andrea, may I hand to you for your thoughts on this wonderful session that we've just heard.
So thank you, Joan. It's a real privilege to be up on stage and see all these people together, to be able to look out amongst all of you and realize that together we built an organization, we built a network, we built a family that has not only discovered, developed, but also delivered medicines which have changed the lives of so many children throughout the world. So I was asked not to talk about the glories, but a little bit to talk about the lessons that we've learned, the problems that we've overcome, and the barriers we've broken. So let me start with four vignettes, four themes to go forward. So the first is bringing in partners from industry and building collaborations. When I first joined MMV, which is, 12 years ago, one of the main questions in public health was, how do you get access to all this diversity that the pharmaceutical industry has? All the mystical drugs that they have, in their cupboards. How do we move things forward? And as Elizabeth and Javier talked about, then screening technology had changed a lot 12 years ago. And so we were able to go to companies and say, we can screen your compounds for you and see what you have. So that's already a step forward. But I think the most important thing was underpinning it, the work that our BD and legal teams did in saying that the intellectual property that came out of that rested with the people that own the molecule. So it became their responsibility to move forward. So at the end of it, not only do we have a few thousand chemical series for Jeremy and his team and all the chemists to start working up, but we have a network of around 30 different companies, each of whom are empowered to work and really believe in the molecules they're working on, because actually they're their molecules. They have skin in the game. Second one is all about data you can trust. So everybody trusts their own data, everyone here. The problem is that we're working in a portfolio where you have to trust that when we have data on somebody else's compound, you actually trust the, how that ranks compared with yours. One of the things that's really magical about the malaria community, a lot of the people sitting behind me, but all of you, is something that doesn't really happen in other therapeutic areas. So several of you, Didier said 15, took on an individual assay which you did for the whole community. Now, that's really special, because what that means is that everybody knows that their compound is being ranked against everybody else's compound compared by the same protocol, in the same way, in an unbiased way. And that means that as we move forward, we actually have confidence as a community in the data, because actually the data were developed by the community. Third thing, and we had a reference to it by James and Jörg, is the development of experimental medicine. How do we know which molecules to place our bets on? We don't have infinite resources, so there's always this question that everybody says, I want to drive every molecule fast as forward as we possibly can. Doesn't always work like that, so we have to pick out the ones that we're really going to push hard. So we built an experimental medicine platform, which, yes, is volunteer infection studies. It's also looking at the clinical data. We've expanded our modeling and simulation, and we continue to learn about that. But also profound deep mining of all the pharmacology data, so that we can say, as soon as we take a molecule into clinical studies, into phase 1, we can sort of tell, is this a molecule that we really want to fast track? Is this a molecule that we still have a few questions about? So we know basically how to put our bets in the right places. And actually, when we get the bets wrong, we learn from that, and we learn where to place them better in the future. So the fourth point is more about the sociology of saying, we love to think that, actually, certainly in R&D, we love to think that, the science and technology questions are the real ones that keep us busy. And that's true. But sometimes the question is much more about solving the most important problem in front of you, even when it's not a science and technology question. So in the severe malaria area, then it's really interesting to watch that, the artesunate injections is actually one of the places where we made the most impact in terms of lives saved. But in the end, the key decision there was actually, first of all, picking the right partner. So I think Fosun, I can't see any of you, but I'm sure Fosun is at the back on the back row somewhere. And also that saying, we're working with organizations to try to translate the real stringency of GMP manufacturing. As Joan said, this is the first time WHO had pre-qualified an injectable. Major achievement. And the main thing was translating that GMP manufacturing into a local context. When we looked at the suppositories, we have a suppository form, which is great because obviously it doesn't need a needle to be injected. It can be used for pre-referral diagnosis. The key thing there, again, was the underpinning the legal framework, the BD framework, which meant that we could actually share information between our various partners freely in a way that somehow didn't seem to happen unless we were there. So we created an environment, and it really does underline that MMV has great science successes, great development successes, but this role as a neutral broker of information is really important. So in conclusion, 3 thoughts. So one, as we've heard so many times, we overcome barriers by passion. I wanted to say that, so many of the partners behind us have been with us over the last decade, and then I look around and I realize that actually we've been pushing these projects along with a lot of these groups, a lot of you. We've been working together with your passion for 20 years, and that it needs that continual investment of energy, resources, and new ideas just to keep things going. Second P is process. Doesn't sound very exciting, but decisions driven by data, and the data we all trust, because actually we all generated it amongst the community. And then finally, of course, partnership. So it's really great to see the way that the community as a whole, when we see a problem, we all come together to try and find a way either over it or around it. So thanks. It's a real privilege to be today with all of this group, but all of you as well. Thank you.
Right, so as Tim was just discussing lessons learned on the scientific side, I will, well, one lesson learned is always to adjust your mic before you sit down. There we go, thank you. To talk about essentially the organizational side of the development lessons learned. If I can just give you a little bit of context, if we can just roll back 20 years, 1999. The Millennium Development Goals are still a year incoming. The Bill and Melinda Gates Foundation won't be formed until next year. The Global Fund won't be started for another 2 years. President's Malaria Initiative out of the US government won't be founded for another 6 years. So for my colleagues who were not in the workforce 20 years ago, which I think is about 60% of MMV at this point, another just small point of history, those of us who were extremely innovative in our technology were carrying around BlackBerries, which had been, in fact, developed that year. So you can imagine that the environment in which MMV was founded was one that was very, very challenging. And I can only imagine, as somebody who has been responsible for the last almost 10 years for overseeing the funding of the organization, the challenges that Chris Henschel and that Peter Potter-Lesage would have felt when they were going to our early donors, to the Swiss Development Corporation, to UK DFID, to not only say malaria is important, but malaria R&D is important, and malaria R&D in a not-for-profit environment is important. It's a huge, huge step and a big risk for the organization. So very, very important place that we founded, and that we stand and we've learned from. A really important inflection point that you've continued to hear during the day around our environment and the external environment then relates to how do we manage our relationships with the external world when we're no longer solely an R&D organization. An R&D organization itself works within a very complex environment, but the limitations of partners and the limitation of government relations is relatively contained. When you begin then to speak about what does it look like when you're talking about patients who are being treated not in clinical trials but actually in the field, what does it mean in terms of government relations? How do you staff this kind of organization? Where do you move with this? An enormous learning for MMV, and one that now that we have multiple products and hundreds of millions of treatments, one where we really are beginning to be able to share our learning moving forward. So the third important inflection point, I would say, that's really changed the way that we think about ourselves as an organization was, in fact, with the movement towards the SDGs. I think for a long time, MMV stood really true to its roots and its early moments where we just were minding our knitting and we were staying close to home and making sure we were thinking about malaria, only malaria, and this is where we were going to work. What we began to realize at the inception of the SDGs is that it is not only malaria and it is not only R&D where we need to be thinking and working and influencing and acting. And in particular, the importance of making partnerships with other product development partnerships, with the TB Alliance, with FIND, with DNDi, and moving ourselves to make sure that the environment in which we are operating is one that we're going to be able to continue to operate in for a long time, and that we can influence and share where we need to. So in closing, I just want to say that, that MMV honors those of you who started this organization, and we thank you. And for those of you who are going to continue this work, both within the organization and moving forward, we don't know what you're going to have to learn. We don't know whether or not mass migration due to climate change is going to change the way that malaria affects us. Frankly, you may look in the next 20 years very, very quaint, having typed away all of your information with your thumbs when in fact it's been embedded in your brain, or the artificial intelligence is going to change it. So we wish you, moving forward, the greatest of luck, and we thank you very much. Yeah, we do. Okay. And so Tim and I have just cooked up something ever so slightly insolite, as they say here. We recognize that within our audience are two real heroes of global health, both of whom have their birthdays today.
Yeah. So we have Professor Sir Brian Greenwood, who, just like MMV, is 20 today. And I think we have Bernard as well.
Bernard, we think we're here. You're here.
I think he's also 20.
Are you here? Yeah. So we're about to go to a coffee break. But since I'm American, I like to sing happy birthday. It's no longer under copyright. Do you mind if we sing a happy birthday to these truly remarkable gentlemen?
Go for it. 1, 2.
Wow, 3 heroes of global health. Tim did say that statistically there must be someone. So if we could sing happy birthday to our elders, okay. And ready?
Okay.
I'll start. Happy birthday to you. Happy birthday to you. Happy birthday, everyone. Everyone. Happy birthday to you. And I think we have coffee break now, Jen.
Yes, thank you, Andrea. Thank you, Tim. Thank you for everybody in good voice. It's now time for coffee. We have about 45 minutes for coffee, networking, catching up with friends old and new, and please be back here at 11 o'clock.